You are most welcome to this talk. It's Monday the 14th of February. Now, sooner or later, we're going to have to change strategies and go for natural immunity. We can't keep vaccinating people all the time. Sooner or later, the question is, when is that? And this is a question that hasn't really been addressed by mainstream media, and it doesn't seem to have been addressed by government agencies very much either as of now. So what I'm going to do today, and it's a fairly academic talk today, I'm going to give a lot of evidence that points out how efficient and effectively natural immunity works. So when someone's exposed to the virus, that's when they're exposed to the antigen, they get a natural immune response, they get an antibody response, and they get the cellular response, the B and the T cell response, and other aspects of immunity we don't fully understand. At least I don't fully understand. And the immune system is incredibly complicated. And as well as that, when someone's exposed to natural immunity, the virus is going to go in through the respiratory tract. And we learned from Professor Clancy that this generates mucosal compartment immunity, that which is in the mucous membranes, these tracts in the respiratory passages that are lined with mucus that go all the way from the nose all the way down to the tiny airways, that this generates the level of immunity as well as the systemic immunity. And of course, natural infection gives this natural mucosal immunity. And this is effective, and we know this is effective, so I'm going to give some evidence for that. Now, I'm largely basing today's study on this paper here from The Lancet. But just to show you, this is why I've been a bit distracted with this for the past couple of days, that there's a lot of evidence here. Now, one of the great things about this is all of the major journals are still allowing you to download this material free of charge, which is actually pretty good. I mean, they should do this for everything all the time anyway, because most of the research is paid for by the public anyway, who fund the universities very largely. But so it should be like this for everything. But it is like this. It is like this for COVID. So, but largely based on this paper here from The Lancet. Now, I'm just going to give you a quick sort of idea of some of the literature we've looked at here. So there's going to be this paper from Science about immunological memory, then this one from Science, ultra-potent antibodies against diverse and highly transmissible SARS coronavirus 2 variants. This article here, this one here, this one here. I'm not going to show you them all now, but these are all trust me, all these are referenced, and I've checked all the references to all of these evidence sources, because if we're not evidence-based, we might as well pack in and go home. So all of these articles here are the evidence base that we're going to be discussing. And if you wanted to spend a couple of days doing this, you could actually go through, you could easily go through all of these and check them out, because I always provide all of the links. It's taken me a while to go through it, but worthwhile, because this is the way ahead. You see, Omicron, I believe, has fundamentally changed the risk-benefit analysis between natural infection and vaccination as the way ahead. That's why I wanted to be quite fastidious in how we give evidence for this. So let's launch into this now. So natural immunity. Natural infection-induced antibodies leads to lower rates of infections, and not just antibodies, the memory cells as well. Now, the questions here, according to this paper here, this paper that we're basing at, it's not really a meta-analysis, it's more of a literature review. But it goes through some very poignant literature on it. And the other thing about this study is the authors spent a lot of time looking through, and they only included what they thought were good quality papers. So all of those papers we've looked at, according to the adjudication of these authors, were good quality papers and worthy of citation. And the degree to which I've been able to look at them over the past couple of days, I must say I concur with that. Not all peer-reviewed, mostly peer-reviewed, not all peer-reviewed yet, because the peer review process takes time. But we'll point out where it's not fully peer-reviewed as of yet. So the question is the strength and the duration of such protection of natural immunity. How long does it last compared with that from vaccination? And the evidence I'm accumulating basically is showing that natural immunity is beating vaccination hands down. Largely because it's a polyclonal response, and largely because it seems to last for much longer. So all those papers there, we'll leave that for now, because I want to show you something else. Let me just show you a quick clip here from the doctors we talked to in Uganda.
In the one of the journals, and they were saying that the Omicron variant is the vaccine that we failed to make.
Yes.
In other words, everyone is getting it.
Is getting it, yes.
Not having any serious illnesses, but it's boosting the antibodies
yes.
To fight any other kind of COVID-19. So in other words, I think in the
there you go.
One of the journals.
We did look at that quite extensively at the time. Omicron is the vaccine we failed to make, and it's boosting the immune response. And what he's saying there is just so obviously true. It's just so obviously true. Anyway, back to the evidence. So this paper looked at PubMed from the inception of the pandemic through to September 2021. They were able to go through, and they looked at biological, epidemiological, various types of evidence. So biological evidence, epidemiological evidence, clinical evidence. Previous COVID infection reduces the risk of infection, which it does, and reduces the risk of hospitalisation. Now, they looked at biological studies first of all, and always. So what I've done here is I've given you the authors, I've given you the journal it's published in, and we've given the title of the journal, and that is the web link there. So I've done that pretty well all the way through. Not quite at the end, but it just shows we've been quite fastidious about the way we've collected the evidence here. Now, this study here says 95% of patients, 95% of participants tested retained immunological memory at about 6 months after having COVID-19. So immunological memory going through 6 months, but this refers to the time when the study was done. It could well be longer than that. This particular study showed more than 90% of participants had CD4. So these are the T helper cells, CD4 plus T helper T cell memories at 1 month. Well, obviously they'd have them at 1 month, or well, not really. So they kind of developed after that time. This doesn't develop as quickly as antibodies, and 6 to 8 months after. So again, 6 to 8 months was the limit of the study. So we've got good cellular response at 6 months, 6 to 8 months according to this study. Next study, again published in Science. Ultra-potent antibodies against diverse and highly transmittable SARS coronavirus 2. Now, one of the things that's coming out really quite strongly in this study I've been doing here of natural immunity is this. So most of the vaccines, like the vaccines we've been using in Western countries, the adenovirus vector vaccines and the mRNA vaccines, they're just giving us protection, helping us to recognise the spike protein as foreign. But if you have the whole infection, of course you have the whole virus, so you can make antibodies and T cells to the spike protein, to the nucleocapsid protein, which surrounds the actual RNA inside the virus. You can make antibodies to the, I don't know if you make antibodies to this one or not, but you certainly make antibodies to the envelope proteins, the membrane proteins. And I don't know about the viral genome either, but certainly these ones. In other words, and plus there's about 22 others that we haven't put on here. When we looked at this, there was 22 different so-called epitopes, the parts of the virus that the immune system recognises being foreign. So you get this very polyclonal response. So anyway, this paper said previous SARS coronavirus 2 infection with an ancestral variant produced antibodies that cross-neutralise emerging variants of concern with high potency. Now, this hasn't quite fully been borne out by history because we know more, because remember this goes back to 2021. What we know now is that infection with Delta does not provide good protection. So infection with Delta does not provide good protection against Omicron. But we do know that Omicron provides good protection against Delta, which is one of the reasons that it's gone extinct. So Omicron does seem to have almost sort of reset the situation to give us this high level of immunity again. Moving on to epidemiological studies. Assessment of protection against reinfection with SARS coronavirus 2 amongst 4 million PCR tested individuals in Denmark. So I think we can call that a fairly large-scale study. People who had COVID-19 previously were around 80%, 80.5% protected against reinfection. Now, again, this does depend on the time when the study was done. Again, this is 2021, and it does vary depending on the variant. But as we say, Omicron does seem to have this back protection against the previous variants. But they were showing 80% protection, which is pretty good. And another study, where were we at? Another study here. This one's in the European Journal of Clinical Investigation in Austria. So people who had COVID-19 previously around 91% protected against reinfection. So again, pretty good. And a frequency of hospitalisation due to a repeat infection was 5 for 14,840 people, which was 0.03%. So we're seeing good levels of protection against hospitalisation. And in all those people, in terms of reinfection, in the cohort they were looking at, there was one death, 0.01% from that particular study. This study here, reinfection rates amongst patients who previously tested positive. This was a retrospective cohort study in the United States. People who had COVID-19, again, previously were 81.8 protected against infection. So we're seeing this 80 to 90% figure really quite consistently. Again, Omicron is more infectious than the other ones. There is more reinfections with Omicron. But I believe it's also generating higher levels of immunity. Necessity of COVID vaccines in previously infected individuals. This is a question mark. So is this necessary? This was a retrospective cohort study in the United States. And the answer here is people who had COVID-19 previously were 100% protected against infection against that particular study. So that one was even more impressive. Comparing SARS coronavirus to natural immunity and vaccine-induced immunity, reinfection versus breakthrough infection. This was in Israel. People who were non-vaccinated people were 13 times increased risk for infection with Delta compared to those who had no COVID-19 previously. So this is nothing to do with vaccination. So if someone was unprotected compared to someone who had the infection previously, regardless of vaccination status, previous infection meant they were 13 times less likely to become infected. So 13 times. And again, we can see that represents a very high percentage. Incidence of severe acute respiratory syndrome coronavirus 2 infection amongst previously infected with vaccine, previously infected or vaccinated employees. So this was an occupational study. Laboratory staff routinely screened for SARS coronavirus 2, so people working in laboratories. People who had COVID-19 previously were 100% protected against reinfection. So again, pretty high level of protection. And we'll just have a quick look at the limited data that's available from the States here. This paper here, this is the this is the let me show you. Here we go. Rates of incidence of large confirmed COVID-19 cases. So these were the people that were vaccinated. These are the people here that are unvaccinated. Because this is higher risk at the top. That's right. High risk at the top, low risk down here. So people that were unvaccinated, high risk, of course. But people that were vaccinated, lower risk. But people who'd had natural infection, way lower risk. That's the line for people that had previously infection, even if they weren't vaccinated. In fact, this line was not vaccinated. This line had previous infection and were vaccinated, and we see they're about the same. So previous infection from this data is giving comparable levels of protection, whether you're vaccinated or not. And the same was actually true for hospitalizations in this study as well. So this one was hospitalizations. And again, we see high risk here for people that were unvaccinated, this line here. But then we see much lower risk for people that were vaccinated. That's that thicker blue line there. But then the thin blue line is people that had previous infection but weren't vaccinated. And again, the darker line, people that had previous infection and were vaccinated. And again, we see virtually no advantage to vaccination on top of natural immunity. So this is just so obvious what we need to be doing here. We need to be testing people, and if they've had natural infection, this data has shown there's no advantage to vaccinating them. And we need to be doing more of these tests. So instead of rolling out all these millions of antigen tests now, which basically are irrelevant because the virus is everywhere anyway in the days of Omicron, we need to be testing for antibodies and evidence or evidence of previous infection. And if that's the case, this data is showing there's no advantage to further vaccination if they've already had the infection. We're doing a one-size-fits-all at the moment, and that goes against everything I understand about individualised medicine. Clinical studies. I won't bother going through all these. You can read them in detail if you want to. But this is the Siren study. So this is actually, I will go through this one. SARS coronavirus 2 infection rates of antibody positive compared with antibody negative healthcare workers in England. So this is a very large scale study. A lot of my friends and colleagues have been involved in this. Large multi-centre prospective cohort study, previously diagnosed of COVID-19, 84% decreased risk of infection. Now, this is just a huge study, and that's pretty definitive data. Another study here, SARS coronavirus 2 seropositivity. That's whether they're testing positive for antibodies, and subsequent infection risk in healthy young adults. Prospective study. I think it was done in US. Yeah, this one was done in US Marines. Again, 82% protection. So we're seeing this again. And of course, they didn't get as sick, and they didn't get sick for as long. As we say, Omicron people are getting reinfected more, but the illnesses are overwhelmingly milder. Again, reinfection here, this was 9,000 people in this study, and the reinfection rates were only 0.7%. So even more protection. So what are we saying here? Risk of repeat SARS coronavirus 2 infection decreased by 80% to 100% among those who'd previously had COVID-19. Studies were large and conducted throughout the world, and again, as we said, the paper picked good quality studies. Protection from reinfection is strong and persists for more than 10 months at follow-up. And we've already given the reference for Hansen et al. And again, of course, as time's going on now, this time period is still extending. Now, this paper published in Nature, SARS coronavirus 2 infection induced long-lived bone marrow. So long-lived bone marrow plasma cells. So the plasma cells are the B cells. These are memory cells. So these are the B cells, the B lymphocytes. And why the B lymphocytes important? Because these are the cells that actually produce the antibodies. So the memory B cells could produce lots of specific antibodies, these Y-shaped molecules, in very large amounts in very short notice, as stimulated by the CD4 cells, which are the T helper cells that stimulate the B cells to do that. And all the while, we've also got these CD plus 8 receptor cells, which are the T cytotoxic cells, which will directly kill virally infected cells. So we've got these different arms of the immune system, and they all seem to be primed, and they can be identified. Now, clearly, you don't want to be taking bone marrow samples of everyone. Quite an invasive procedure. Patients don't like getting bone marrow samples. Usually, you normally have to stick a needle into the sternum or into the pelvic bones. It's quite an invasive procedure. That's why we haven't got a lot of data on it. It's not just like taking a blood sample. A study amongst post-infections individuals who experienced mild SARS coronavirus 2. Mild infections induced a robust antigen-specific, long-lived humoral immune memory. And it's a long-lived humoral immune memory. Humoral, of course, means it's in the fluids of the body, and that's exactly where these antibodies circulate, in the body humours. So that is cellular immunity generating the humoral immunity. Now, this study here, this is really quite important. T-cell response to SARS coronavirus 2 infection in humans, a systematic review. SARS coronavirus 2 infection induces specific and durable T-cells, which has multiple SARS coronavirus 2 spike protein targets or epitopes, and as we looked at on that previous diagram, as well as other SARS coronavirus 2 target proteins. So again, we see that the natural infection, I mean, just look, here's a picture of this virus. This is the public domain one from Wikipedia, and the complexity of it's incredible. So all those different molecules on there which can be inducing an immune response, and that's just the outside, of course. We can't see the ones in the middle. So we know that in the middle we have these other proteins that we've shown that will also generate the immune response, as we saw from that very simplified diagram there of the other components. See, all of these components aren't human, therefore they'll be recognised as foreign. They're all non-self components, obviously. So broad spectrum polyclonal. SARS coronavirus 2 specific T-cell immunity in cases of COVID-19 and SARS and uninfected controls. People who recovered from SARS coronavirus infection in 2002-2003. So this is what we could call the SARS coronavirus 1. The people who'd had that in 2002-2003 continued to have memory T cells that are reactive to SARS coronavirus proteins 17 years later on. Maybe that's the most profound thing I've said today. So people that had SARS, severe acute respiratory syndrome, in 2002-2003, when this study was done in 2020, they were still generating an immune response to the original SARS coronavirus, what we could call type 1, 17 years before. So the immunity lasted for 17 years. Let me just say, I don't expect vaccine immunity to last for 17 years. Does this mean to say that it will be the same with SARS coronavirus 2? Of course not. But it's pretty promising that that's how long. And of course, SARS coronavirus 1 now no longer exists. It's not there. But we do know that people that were infected with SARS coronavirus 1 have got good levels of immunity against SARS coronavirus 2. So again, pretty promising because they are different viruses. Memory B cell response to SARS coronavirus 2 evolves between 1.3 and 6.2 months after infection. So the fact that what you find is that when the cellular response is delayed by several months after active infection, that tends to bode for longer term protection. So epidemiologically, we've got this indication of longer term protection from natural immunity from the SARS coronavirus 2002-2003, and also from the biological perspective, because the cellular response is taking longer to be generated with other infections, that means that the immune response will likely last for longer if it's consistent with other infections. And to be quite honest, why should it be any different? Some people who've recovered from coronavirus might not benefit from coronavirus vaccination. That's putting it really quite mildly. That's that paper there. Not peer-reviewed, it's a preprint, but that's what that's saying. Some people who have recovered from coronavirus might not benefit from coronavirus infection, which of course is 100% consistent with this data from the States. There's limited data here, so people way down here who've had the infection, basically that line is on the same trajectory as people who've had the infection that have been vaccinated. So would you want to be on the solid black line, or would you be on the dotted blue line? What doesn't make much difference really, doesn't it? So that indicates that the vaccine wouldn't make much difference, as the natural infection would. One study found that previous COVID-19 was associated with increased adverse events following Pfizer vaccine. Okay, so that's saying the vaccine could actually be doing harm in people that were infected. And this has led to the thinking in Switzerland. In Switzerland, proof of recovered infection, so you've had a test, you've been diagnosed, you've recovered, in the past 12 months are considered equally protected as the fully vaccinated. And of course, this is completely correct, as the CDC's own line shows. It's the same. It's essentially the same level of protection. That's the protection against hospitalisation. That's from the California one. And the New York data that shows the protection against hospitalisation, again, if you've had the infection, the vaccination gives no further benefit. So they seem to realise this in Switzerland, but in my country, we're not bothering to differentiate. And I use that phrase quite deliberately. We're not bothering to differentiate our vaccination strategy for people that have been infected naturally or not. We're not bothering to do that. And this goes against everything I understand about individualised medicine, as I've said. So that's what we're doing in Switzerland. Right. Direct quotes from the paper. Although longer-term follow-up studies are needed, of course, you always say, but I don't know if you should be allowed to say that, really. When I was marking a lot of academic essays, we actually had a rule that says students are not allowed to say that more research in this field is necessary. The research we've got is the research we've got. Of course we're going to know more next year, in 50 years' time, in 100 years' time. Of course we are. It goes without saying, but anyway, that's... Why did they bother saying that? It's so evidently true. Anyway, clinicians should remain optimistic regarding the protective effect of recovery from previous infection. According to these authors, policymakers should consider recovery from previous SARS coronavirus 2 infection equal to immunity from vaccination. That's what the data is showing, but we're not bothering to differentiate. So remember what the Ugandan doctor said, Omicron is the vaccine that we failed to make. And for those of us that did manage to make vaccine, has Omicron made vaccination obsolete? If you've been infected with Omicron, has that made vaccination obsolete? Is a pressing question that we really need an answer to from Chris Whitty, Patrick Vallance and the CDC and all these guys really soon. You get these people in the BBC. We have these, or any mainstream media really. We have these briefings for the Prime Minister, and they get in to ask these questions. So they actually get the Prime Minister. They get Patrick Vallance and Chris Whitty and all these people there, and they get to ask these questions. But because they're not scientists and doctors, maybe they don't ask the most penetrating questions. So is our mainstream media failing to ask the most penetrating questions of our politicians and scientists? Is another question I will leave you with. But that's the penetrating question. We must have answers to this soon. If someone's already been exposed to natural infection, should we still be vaccinating them? And we really need a question for that, because all this evidence is showing that the natural immunity is really pretty good. Okay, thank you for watching.